Okay, so we proceed to the last talk of the session. That was Dr. Abhijit Majumder is as assistant professor at the Department of Chemical Engineer in Indian Institute of Technology, Bombay, and the Wellcome Trust DBT India Alliance Early Career Fellow. His research group focuses on different mechanical aspects of a tissue influence cellular behavior and fate, as well as attempting to mimic cells in vivo environment. He is the author of several publications in international reputed journals, including Science, and received several international and national awards, including People Travel Grant Award, Alan Gant Runner-Up Award from Adhesion Society USA, and the TR35 Young Investigator Award by the Technology Review MIT. And he will present us today his work on mimicking glioblastoma, importance of mechanobiology.
Thank you for this nice introduction, and thanks to all the organizers to give this opportunity. I have interacted, with many of you in last few days in the workshops, and I think, it feels really great that finally to be in some in-person meeting and finally get the chance to meet people face to face, not, behind the scene, behind the screen. So I thought that, last few days when I was, interacting with many of you, and I got this kind of questions that, are you one of the organizers? Are you working on organ-on-chip? Are you working on organoid model? So I thought that before I get into a complete identity crisis, let me first tell you what my lab works on. So we call our lab as AIM lab, and AIM stands for Materials, Mechanics, and Microfluidics. So these three, as we think of as our base. So we think of to have different materials. Then we would like to understand the mechanical properties of those materials, and then we would like to understand how the mechanical property of the materials may influence cellular behavior. So this field is called mechanobiology, and you got a little bit of glimpse in last two talks. And the third part of our lab is microfluidics. There we try to make different types of microfluidic devices. With these three base, we try to understand the role of mechanics in stem cells, cancers, and other relevant cell biology problems. And then, with all this kind of putting together, we try to make a few different kind of devices, which our main goal is to make something that is cheap, affordable, but still kind of, something that, many of the labs in India or in our kind of countries, as we all know that the many of the labs are resource constrained, as Karishma also kind of pointed out yesterday. So that's something that they can do and they can use. And so this, high throughput spheroid generator and the drug testing device, these two we are also covering in our workshops. I hope that you can take it back to your lab and you can do it yourself. But we also work a bit on the various different organ-on-chip models. So these are the three things that we are working on. Placenta-on-chip, for which Anshul has a poster outside. So this I am doing with my collaborator, Dr. Deepak Modi from NIRRH Mumbai. Skin on chip and the retina on chip, these two I am working with Dr. Prajakta Dandekar from ICT Mumbai. You have heard a talk from Devashri yesterday or day before yesterday on retina on chip, and for skin on chip, Deepa has a poster outside. So all of these various things you will get to see outside when you talk to them. My student Saurav will be, he has a poster on a spheroid making device. Sheetal has a poster on how we can mimic from available template, and you can pattern cells, because cell patterning also an important aspect if you try to mimic what is happening in vivo. And Ketki will talk something that, Ketki will have a poster outside that is essentially an extension from my talk, where I will stop. From there, Ketki will start. So this is just to give a little glimpse of what my lab is working on. And so the next slide, I don't think I need to even talk about this slide after these 2.5 days of discussion, that we need to mimic, not an alternative to animal, as someone has pointed out. We need alternative to human predictive model, right? But so far, up to this point, we have talked about all 3D or organ-on-chip and the organoid 2.5D, 3D, that kind of models. Now I'm going to talk about 2D models, odd man out, black sheep in this room. But then, why I'm talking about a 2D model in such kind of meeting? So I would like to stress about a point. I think someone in the morning mentioned, I am a mechanical engineer and I want to know about the effect of the, yes, that guy is sitting there, effect of the mechanical forces. So I would like to talk about, even in our 2D culture, how the mechanical properties is important, and how by mimicking the mechanical properties of the native tissue or native organ that I am trying to mimic may give us a better model, better mimicking model. So I will try to explain that using a specific example of our work, that is mimicking glioblastoma, and I will try to talk about importance of mechanobiology in that particular context. So this work is done from my group, Pallavi. She has graduated last year, and now Ketki is kind of continuing that work. And this work is in collaboration with Dr. Shilpi Dutt from Actrec and her student Angha. If I just say the collaboration, I will not be very truthful. The thing is that in just this a similar kind of meeting, after my talk, we were having lunch together, and then Shilpi told me, see Abhijit, this is a problem that we are seeing. She described something, I will just talk about that. And then I said, okay, let us think from the mechanical point of view. So I stepped into the glioblastoma thanks to Shilpi. So it is not a 50-50 collaboration, 80 goes to Shilpi, maybe 20 or even less than that with me. So why glioblastoma? So as I have already noticed that many of you working here in the field of glioblastoma, so it is one kind of brain cancer, start from the glial cells, and it is the prognosis is not good. Okay, if one gets the glioblastoma, for sure that the end is not far away. But more than that, I would like to draw your attention to these two curves. One is from 1967, another is from 2014. Expectancy, survival over year or over months. And you look at carefully that from last 50 years, for this particular cancer, we actually have not progressed much. We could not crack this nut, right? And even with surgery, radiotherapy, chemotherapy, with everything, life expectancy is 1 year to 1.5 years or so. But one interesting thing to note here is that when the patient has the primary tumor, I treat the primary tumor with, say, radiotherapy, chemotherapy, surgery, whatsoever. The tumor relapses, comes back after few months, 6 to 8 months. And it has been shown that the relapsed tumor is much more aggressive than the primary tumor. And what do I mean by it is more aggressive? It is highly proliferative, more tumorigenesis, more invasive, more migratory, and drug resistant. And then the patient dies. So actually the patient is not dying of the primary tumor, patient is dying of the recurrent tumor, right? So we need to really look at the recurrent tumor carefully. Problem is that we really don't have a coupled sample, that from the same patient I have a primary tumor sample as well as I have a recurrent tumor sample. Because when the recurrent tumor happens, it is already invasive, and so the surgeon does not really prescribe to go for a surgery. It is meaningless. And in very few cases, after death, they donate the body so that we can have the samples, right? So there are only few kind of the samples exist, they don't exist. So Shilpi's lab, what they have is that they have the tumor cells, parent tumor cells, they expose them to radiation. What happens is that after they expose them to radiation, most of the cells die. So if you look at your plate will be almost, you cannot see the cells. But if you keep the plate for longer, some giant cells appear, multinucleated giant cells. Then they go through the cytokinesis, they become single nucleus cells. And that is what we are talking about here as a radio-recurrent cells. So in this talk, I will use the word recurrent, relapsed kind of interchangeably, although they are strictly not interchangeable, but still I will kind of do that. What they have done, so in Shilpi's lab, they have these radio-recurrent cells. They looked at their molecular markers, and they have established that yes, radio-recurrent cells matches with the patient recurrent sample, whatever we have. So they have established that model. However, they were facing a problem, and what I was talking about the lunch meeting, is that, so what they have, as I told, say, for example, they are working with, say, cell line U87Mg. We call it the parent cells. This is the residual resistant cells, radio-resistant cells. You got the recurrent cells. You are culturing them on your plastic Petri plates. I already told you these cells are supposed to be more aggressive. But when they take these cells and look at their migration, yet recurrent cells has a higher migration, but the difference is not that prominent. Invasion, yes, recurrent cell has a little bit of extra invasion, but again, not that prominent. So I don't have a model that can capture the aggressive nature of my recurrent tumor. I don't have a model. But when I put these cells in the mouse brain, clearly the recurrent cells are more proliferative, more in number, and also more migratory. So what is happening? So clearly our in vitro plastic petri dish culture is not being able to capture the scene. So this gave us a clue that something is happening in the brain, what is absent in our system. And here we start thinking that maybe the mechanics is playing a role. Because our plastic petri plate stiffness is gigapascal, 10 to the power 10 Pascal, and brain is one of the softest tissue, 500 Pascal. So it is something is that you were sitting in this nice cushioned chair, and all of a sudden I take you and put it in a stone made, someplace. Your behavior will not remain the same, right? So we ask the question is that, can I give them something that they like? So here comes the thought process of the mechanobiology. So it is already known and established using various other cells that depending on the substrate stiffness, cellular behavior changes, various cellular behavior changes. And how do the cells senses the stiffness on which they are? And I am talking about the adherent cells. So they attach to the substrate, then they apply the traction force. You have heard of it today. They apply the traction force, they deform the substrate on which they are sitting, and by that deformation, they can understand whether they are sitting on a soft or stiff, and based on that, they behave. So we are not the first one who are looking into the effect of stiffness for the glioblastoma. It was already known, and other peoples have shown that the glioblastoma cells, GBM cells, can sense substrate stiffness. So that is established. But our question here is not whether the GBM cells can sense substrate stiffness. Our question is also not to, understand that how the mechanical, property affect the GBM. Our question here is that parent and relapse, how they react to the mechanical forces. Why again we are thinking of this? Again, when we looked back at Shilpi's data, they have done with 2 cell types and 1 patient sample. They have compared parent with the relapse, and you can see that, so there is the 2 cell lines and 1 patient samples, various different pathways, and they are comparing parent with relapse for, they have the different things. But this set is common for all these 3. And out of this set, one is obviously pathways in cancer, but other is focal adhesion and ECM receptor interaction. So again, that tells us ECM has something to do with what we are seeing, the difference between parent and relapse. So we are asking this question: do the parent and retrorecurrent cells, they behave differently in their mechanosignaling, mechanotransduction, mechanoresponse? And second question, can we recapitulate the aggressive nature of the recurrent tumor in vitro by mimicking brain stiffness? So these are the 2 questions that we tried to answer. What we did, so this is what I have already told you. This is what Shilpi's lab was doing that point of time. What we got into the picture is that we made the acrylamide gel of 0.5 kilopascal, 500 pascal, mimicking. So we took the mouse brain, we measured the stiffness using rheometer, and we made this gel. This gel is coated with collagen so that cells can adhere, and then we put the cells on top of it. So just throughout the next 20 minutes or so, please remember this. I have this 4 population: parent on plastic, recurrent on plastic, TCP tissue culture plastic, parent on gel, recurrent on gel. These are the 4 populations I am trying to compare. Our first question was that, do they have a difference how they sense the substrate? One of the first thing to look at is the cell spread area. On stiffer substrate, cells spread more. So I have the cells parent and recurrent on 0.5 kPa, 10 kPa gel and plastic. Just if you look at these 2, probably you don't see much difference. They kind of look very similar, parent and the recurrent. But when we look at the cells on gel, parent, you can see all are the round cells. In the recurrent population, a good population was elongated. So a population in the recurrent cells only is coming up, and it is coming up only when you are putting them on soft gel. So it is captured from the cell spread area as well. So if I look at the circularity, essentially what I was telling here, so circularity is 1, meaning it is completely circular. So in a parent cell it is just 1 population, but in the recurrent cell you see another population here which has a low circularity, meaning they are elongated. So clearly they show difference when I put them on brain mimicking substrate. Okay, this is fine. So next we wanted to, so because our question was that whether they have a difference in mechanical signaling, mechanosensing, so the key or the heart of mechanosensing process is cellular traction. As I said, cells apply traction, they deform the substrate, that's how they sense the substrate stiffness. So we wanted to know, do they apply different amount of force on their substrate? So what we did, it is called traction force microscopy. So on a gel, you have a fluorescent beads on the top. You put the cells deform the substrate. You take an image of the beads, then you kill the cell, the gel, the substrate get relaxed. So the beads go back to their original position. And then you take another image, and comparing these 2 images, doing some mathematical jugglery, you get that how much force cells were applying. So what we found here is that, yes, when I look at the just kind of the 0.5 kPa, the combined, parent cells are applying more traction than the relapse cell. So relapse cells are applying less traction on a softer cell. And I am just not going into these details, but this clearly tells us again that yes, the mechanosensing property of the parent and the recurrent cells are different, and this difference can be captured only when you put them on a brain mimicking substrate. Fine, but so what? Our main goal was to capture the aggressiveness. So next we get into that question, can we recapitulate the aggressive nature? So what are the aggressive nature? Proliferation, migration, invasion, tumor formation, drug resistant. First, migration. On plastic, parent and relapse have the same migration. When you put on 0.5 kPa, then your relapse shows much higher migration, close to 80% or than the parent cells. So recurrent cells are more migratory. And this extra migration actually coming from those elongated cells, not from those circular cells. So this population is actually causing the extra migration. Then we looked at invasion. Invasion done was using the Boyden chamber. You probably all know that we took the cells from plastic as well as from our gel. We put them, so here there was matrigel, we put the cells here. There was chemoattractant, and we wanted to see how many cells are invading. Now I am looking at plastic and 0.5 kPa, only parent. Yes, 0.5 kPa, cells grown on 0.5 kPa, when I check their invasion, they are more invasive. Parent population. Now you look at the relapse population. Bang. They are like, invasion increases like crazy. That you don't capture in your plastic culture. Okay, invasion, migration. What about, so we see that yes, more migratory and more invasive. Then we looked at that, so if they are more invasive, what is happening to their MMP2s and MMP9s? That is the MMPs which kind of cleave the matrix for invasion, and we are comparing the recurrent with the parent. Again, on the stiffer matrix, the differences are negligible, but on 0.5 kPa, you can see MMPs are high from the recurrent cells. Interestingly, collagen is also high, meaning is that these cells are actually depositing collagen to create the track, but then chopping also to create their path. And it is known in various different cancers that yes, they do so, that we can capture here for the recurrent cells. We have, so if they are depositing collagen, then the stiffness should increase. Then we looked at again the mouse brain. We took our cells, put it in the mouse brain, created the tumor. We measured their stiffness, and yes, cells, when we create the tumor from the relapse cells coming from 0.5 kPa, that tumor is much stiffer compared to the other tumors. So they are depositing collagen, making their surroundings stiffer. What about the proliferation? Again the same thing, plastic, stiff substrate, not much difference. On the softer substrate, difference is quite significant. This part I would like to draw your attention. This is a very important thing. We are looking at the drug resistance. Sorry for the spelling mistake here. We are using temozolomide, the most common drug to treat the glioblastoma. You can see here, first look at these two. Parent relapse cultured on plastic shows equal resistance. Parent and relapse cultured on gel, huge difference. That is, these cells are drug resistant. So if you are starting with a molecule, and if you test on your plastic, you will say, yes, this molecule is working. But this will tell you, no, it is not working. Okay? And so that is what we see. So essentially we could capture most of the signature of aggressiveness of the tumor. What next? Next is obviously to put them in the mouse brain, look for the tumor development, and again the same thing. Plastic parent, plastic recurrent, tumor, a similar type of tumor, not much big difference. 0.5 kPa, 0.5 kPa parent, recurrent, recurrent gives you much larger tumor than the cells coming from the plastic. So, relapsed tumor cells grown on brain-like gel show more aggressive phenotype. We able to recapitulate all these various different phenotypes that is seen in the clinical evidences. Is our system recapitulating the actual in vivo, the clinical state? I don't know. I am not even claiming that. That's a much bigger beast to deal with. I am saying only this much, that yes, I can recapitulate the phenotype by using my mechanobiology tools, which could not be captured when those cells were grown on plastic petri plates. Well enough. What next? Next, we obviously wanted to know, can we do something? So EGF receptor that has been thought of a long time, a player in the glioblastoma, and then EGFR inhibitor afitanib has been used to treat, but the success rate was low. What we see here, so for the afitanib treatment, it kind of delay, but not much success. What we see here is that afitanib treated cells, yes, invasion go down. That is, it is reducing the invasion, but it is not helping in terms of cell survival. Right? Sorry, here. So the afitanib treatment, the EGFR inhibition is not making those cells more drug responsive. However, it is reducing the invasion. So what to do? So we looked into the differences, that, transcriptome analysis, that what is special in the recurrent cells grown on gel. And we got this whole list, and out of that, pleca-7 that came at the top. So pleca-7 is one of the adhesosome junction protein, adherine junction protein. So it works in the cell-cell adhesions. So then we wanted to downregulate pleca. But before that, we looked into the samples. Here we looked into the staining, and we can see that compared to the plastic cells grown on 0.5 kPa, yes, pleca is high. But it is for the U87mg, sorry. High, but it shows non-significant. But when we look at the patient tissues, then yes, pleca-7 is indeed higher in the recurrent cells. Similar thing we see in the RT-PCR, but yeah. So we looked for the, went for the pleca-7 knockdown in Shilpi's lab. So they downregulated pleca-7, and as you can see here, with the pleca-7 knockdown, the invasion goes down. That we also saw in case of EGFR inhibition. But what was not there in the EGFR inhibition, that the EGFR inhibition did not have any effect on cell survival. But here we can see that when we have my pleca-7, we downregulate pleca-7, we see that the cell survival goes down, meaning it is helping in killing my recurrent cells. Have we found a novel target? I am not again claiming that. It has a long shot to say that. But what I am trying to say, at least so far, is that just being in the 2D culture, I am not going into 2.5D or 3D, but just being in the 2D culture, but thinking of what tissue you want to mimic and how my in vitro culture is different from what I am trying to mimic may give you a step forward. Here we change the substrate stiffness because my TCP, my cell culture, plastic and gel had a huge difference in stiffness. In other case, you may want to incorporate shear stress or something else. But it is always important to remember what tissue I am mimicking and what are the main feature in which, I am kind of going away further. So this is the kind of conclusion that we compared all these 4, and we could recapitulate that yes, the recurrent cells cultured on gel, on the 0.5 kPa gel shows all the signature of aggressive tumor. We could find out one target which might be helpful in the further, going further. This is just a last but one slide. So this is a teaser because, as I said, so I started that I'm working on 2D and not going to 2.5 or 3D. But then the next step is obviously to go to 2.5 and 3D. So that is, Ketki is doing, her poster is outside. So there, now we are trying to do that, we put the parent and the recurrent, and when you make the spheroid in general or tumoroid, we use the ultra-low adhesion plate, we use the centrifugation, etc. But in vivo, the tumoroid or those things, they don't form like that. They don't experience any external agency. It is a self-assembly. Yesterday also one speaker was talking about that self-assembly. So what Ketki is looking into, how these, the cells when grown on soft gel versus stiff gel, how do they self-assemble, and what are the differences we can see in their self-assembly, and whether that can capture some of the in vivo observation. So to know more, I will not tell more, I will not take away the excitement from Ketki. I will leave you here with this suspense and you go to her poster to look further. Last, so obviously, Shilpi, Anagha, Pallavi, Ketki, they all are the main part of this work. These are all the funding agency, and this is how the lab changed from pre-pandemic to post-pandemic. So before I end, just a little bit of advertisement. On 18th November at IIT Bombay, we are organizing this one-day summit on next generation therapeutics. We will discuss about drug discovery, drug delivery, and the regenerative medicine. We have invited in our speakers the policymakers, industry people, and the academia, because what also from there I would also leave you with a parting note that what I realized in last 2.5 days is that just in India, I'm not talking about the overseas speakers, we all are working in various parts of a big picture. Probably if we can hold hands and can bring those various parts together, someone is making a pump. I need a pump. She needs a CFD simulation. Someone is making us a device to study the drug response of the spheroid. Someone is making spheroids. So I think CPHMS also can play, they are already playing, can also play a big role to make a consortium where we all can come together, share not only what we are doing, but also our tools and techniques to each other so that we can grow together. Thank you so much.
Thank you so much for the nice talk and the example of what mechanobiology can do for our in vitro models. So the session is open for questions.
Yeah, myself, Dr. Mamta Mishra from Jaya Research Foundation. It's a so wonderful talk, Dr. Majumder. I'm so happy, and this mechano-sensing property, it opened a new vision for me, because I am a molecular biologist, stem cell scientist, but it opens a new vision in the sense, when I do the culture of the primary keratinocyte or liver primary hepatocytes or brain stem cells, we just take into the surface, do the collagen coating or the poly D lysine. I never looked at this. So rather than question, I need your suggestions. What we should look first, how my cells need, what kind of substrate it need, rather than just going ahead and do the molecular assay. So please suggest us what we should stick to first.
So,
I mean,
first of all, thank you for your kind words. That was really
yeah,
heartening.
really, it opens a new vision. I was thinking what we are doing for a long time
So-
rather than looking at, mechanosensing property.
So as you were talking about the keratinocytes, I will just tell you two things, and I think someone also worked with the mesenchymal stem cells. There was this discussion. So for stem cells as well as your primary keratinocyte, you know that one of the major limitation is that after a certain number of passage, they go into replicative senescence.
Exactly.
You don't get enough number of cells. They also lose their property.
Yeah.
Right?
Yes.
We have shown in our work, that paper is published, if you grow them on a softer substrate, you can delay the replicative senescence, you can increase the cell number. So if you, from your plastic, if you get 1 million cells, on the gel you will get 500 million cells. More than that, another interesting feature is that the optimum stiffness for mesenchymal stem cells and the optimum stiffness for the keratinocytes are not the same. They are different. So what I kind of envision is that when I today go to the company and buy MSC specific media, keratinocyte specific serum, probably we also should think of specific substrate.
Yeah, and one more additional question, like when we think about the growing the primary hepatocyte culture, would you think also the same property will be applicable, and you can give us some suggestion how we'll get the better primary hepatocytes when we are
I
cultured?
strongly believe yes, it will be, but if I say yes or no, that will be a throwing in the dark,
Yeah,
because
of
I
course.
have not done that, right?
Yeah.
But from the gut feeling, I know that it will be.
Thanks
What
a lot.
is the optimum condition, that we need to figure
Yeah.
out by.
Okay, thanks a lot for the wonderful talk. Thank you.
I was going through one of the paper where
Can you just come closer?
so recently I was reading one paper where the authors have proved that when the patient receives chemotherapy, the surrounding cells become senescent, and that senescent cells release certain biochemical cues that make these tumor recurrent and more aggressive. So it's not just the mechanical forces but also biochemical factors that contribute to the recurrence of tumor. So in your system, can you incorporate those cocktail of cytokines or biochemical cues which can further make them recurrent?
Yes.
Is it possible?
Yes, so as you have said, that is absolutely true, and that the senescence associated the cytokines, that has a role to play. So as I said, it is just a first layer of work, right? That I am just played with the mechanism. Yes, I can add the cytokines and the specific cytokines to look that now I can combine mechanical as well as biochemical cues, and actually that will be the real mimicking, because so you are bang on the point that what I was trying to make, that one portion they don't think of the mechanics, they are just thinking of the the chemical signaling part. I will be very, very wrong if I stop thinking about the chemical part, right? Yeah. Those are, we need to bring them together. Absolutely.
Sure. Thank you, sir.
But I think as Karishma or someone told that cytokines are costly, I don't have.
No, for senescence, you don't need cytokines. You can just make them senescent and collect the supernatant. It works perfectly fine. Correct. Because I work on senescence and I have a poster if people are interested in aging and senescence.
Smart.
So, on addition to what Akshat has asked, first question is technical. So, for GBM patients, what they usually give is 60 grays of radiation is the maximum dose which is given in radiotherapy. So, how are you making these cells radio recurrent? Are you giving a single dose of radiation, or is it multiple dose of radiation is how it happens in a physiologically relevant condition?
I think, Ketki, I think single dose of radiation.
Okay.
And I mean, as I said, it was done in the Shilpi's lab, so they have calibrated it.
Yes.
For the cell. Yes.
Understood. Second question is, since we're talking so much about mechanobiology here, you did not show any data on, let's say, actin or the cytoskeleton part of these cells, because the cells are definitely changing the shape, become
Yes.
from more spherical round to being aligned.
Yes.
So do you have a leading edge or some kind of this phenomena occurring in your radio recurrent cells?
So not the leading edge, but I have not shown the data, but as you have correctly pointed out, that the elongated cells, they have a better
Actin fibers.
alignment. Yes.
Okay, thank you, sir.
Thank you.
Hi, sir. A couple of questions. The first is related to the irradiation. So in vivo, normally it would get irradiated after the tumor has already formed and it has already created its extracellular matrix, which may be stiffer than the native brain tissue. In your model, you have taken geometric properties of mouse brain, but not a tumor, right? So do you expect that the...
We have, yes, so just sorry to interrupt. So we had the 0.5 kPa, that is the mouse brain, but somewhere I have also shown the data that we have also measured the tumor. And when we look at the tumor stiffness is higher, and when the tumors are created by the radio recurrent cells grown on 0.5 kPa, those tumors are further stiffer because we see that the radio recurrent cells secrete more collagen.
Right, but there the tumor is recreating its environment. If you were to put it back in an environment that is similar to its tumor microenvironment, would you expect different behavior? Whereas here, you're putting it on a substrate where it has to remodel it and then...
It's remodeling.
So in the brain, that probably does not happen, right? Because it already has a microenvironment and the cells repopulate it. So do you expect the behavior to be different if you use the matrix of a different stiffness, for example?
So if I have understood your question correctly, I'm not very sure if I have understood your question correctly, but when you think of the tumor, right? The tumor first formed. But then when the tumor is forming, that time the microenvironment is like the native tissue, right? The brain tissue, but or maybe some changes, but more or less like the knife tissue. But then they are modulating them, right? They are changing it. So here also, when we are putting the cells initially, we are putting them on a brain-like stiffness, right? And these cells are maintaining that brain-like, because here they can secrete the collagen, but then they really cannot change the stiffness overall, right? So that changing is not happening in my substrate. My substrate is not capturing that. But when I am putting those cells in the brain, they are again starting their process and they are remodeling the brain. So when I am putting my cells on my substrate, the cells are not being able to change that. So, but they are secreting collagen, but it's a 2D substrate. So,
I think I will talk to you separately
probably,
about this.
Yeah.
One more question. So, do you expect, quick question, if you irradiated it on the low stiffness substrate, do you affect a change in survivability?
Very, very, very good question. Very good question. We are also thinking of that. I think there will be some difference, but it will be a throwing the stone in the dark, but it's a very good question. Yes, we are thinking of that.
Thank you.
Thank you.
Thank you, sir, for the wonderful talk. I just have a very small technical question. What was your technical setup for studying migration and invasion in your with your cells, both parental and after the?
So the migration was done using the time-lapse microscopy. That time cells are on their substrate. We are doing every half an hour, we are taking the image. Then using image J, they are doing the tracking. And then from the final to the track length divided by the time is your migrational speed.
So you coated the cells on first coated the cells or like?
No, so I have my gel, right? The gel is coated with collagen.
Okay.
There I have my cells. Cells got adhered to the gel. Say after 6 hours, then I start my live cell imaging. I take image every 30 minutes for 18 hours.
Yeah, understood. I just wanted to ask, like, if you coat it with collagen or gelatin or something.
So I showed that the gel, the polyacrylamide gel has to be coated with collagen, otherwise cells will not adhere.
And in the invasion, what percentage of matrigel did you use?
This you need to ask again the students. This nitty-gritties we don't know.
Okay, thank you, sir.
At least I should not say we, probably other PIs know, but I know, I don't.
Okay, thank you so much. I think now we have a coffee break and then, I don't know how long, and then the poster section.